And now we'll give Jen Morto the chance to reply to what Laura just said. So she will talk about novel genetically engineered mouse model of cholangiocarcinoma. Jen is professor at the CRUK Beatson Cancer Institute, and she has a long-term expertise in animal modeling. Thank you for being with us.
Thank you. I'd like to thank the organisers very much for inviting me, and thank you, Chiara. She's the only one that actually knows I've done any work in cholangio. So that's my first disclosure. I'm not a healthcare professional unless you count mice, and I don't work in cholangio, but that'll become clear as I go through my talk. So just a bit of background, I guess, or what I understand at least about the cholangiocarcinoma models that are out there. Lots of people obviously have used in vitro models, but they can often lack the physiological complexity, obviously not the very nice, elegant models we've just heard about. A lot of the in vivo models involve administering carcinogens or implantation into immunocompromised mice, neither of which are ideal. And the obvious advantage of GEMs, and I should say that I am a GEM modeler by trade, the obvious advantage of them is that you can study right through from initiation, transformation of preneoplastic lesions to invasive and metastatic cancers. So several GEMs have combined oncogenic KRAS and loss of P10, and I'm going to talk about some of our models using those genetic lesions. But those models so far have been complicated by morbidity due to other malignancies, such as HCC, PDAC, stomach issues, and lung cancers. So now I have to give you 2 slides on what I do, because otherwise you'll have no idea why we ended up working on cholangio. So I work on pancreatic cancer. It's the sole focus of my lab. And the workhorse for many years in that cancer type has been the KPC mouse model, which combines conditional mutation of KRAS and P53. And it's a really great model in a lot of ways. It's resistant to chemo, it's got the same genetic lesions as human cancer, it's got a strong fibrotic response. But even as the gold standard, it had a lot of issues. There are multiple cells of origin, there are multiple initiating lesions. That leads to acinar atrophy in the pancreas, which leads to inflammation. When you've got that on a background of oncogenic KRAS everywhere, you get continual, a vicious circle of inflammation, conversion of lesions, and none of these lesions can ever resolve because you've got mutant KRAS everywhere. So we wanted to know what happens if we can just use ductal Cres in the, to initiate tumours instead. This is how we got into cholangiocarcinoma. So we wanted to look at ductal Cres with KRAS due to oncogenic KRAS, obviously, because that's a big driver in pancreas. And as a proof of principle, really, we decided to go with P10 deletion because in our pancreatic models, it's still the fastest driver we've ever seen. So as you can see in the Kaplan-Meier, crazy fast. I can't, does this have a pointer? If it does, I can't. Oh, yeah. Yeah, so if you can see here, that model's crazy fast. So we thought we'll go with those genetics first. So that's what we did. We actually used CK19 CRE-ER and SOX9 CRE-ER. I'll tell you about the CK19 CRE-ER first. So this is the model that we developed, KRAS G12D and P10 loss, induced with tamoxifen at 6 weeks of age, and that's fairly important. So the good news, deletion of 1 or 2 copies of P10 significantly reduced survival in the mice, as you can see nicely by the Kaplan-Meier. And actually mouse models are expensive and time consuming, but this one's pretty rapid. As you can see, 23 days median survival in the P10 homozygous mice. So that was the good news. the good news for you guys, and not so much for us, is that the P10 homozygous mice exclusively developed both intra and extrahepatic cholangiocarcinoma. And you can see that by gross histology, obviously, here. You can even see the pancreas because it's yellow because the mice are very jaundiced. If you look at the ultrasound imaging, you can see essentially progressive restriction of the biliary tract. You can also see that if we look in their bloods, bilirubin's up, ALK-Phos is up, albumin is down. However, the slightly bad news, that's great in the P10 homozygous mice. However, the heterozygous and wild-type mice mostly succumb to lung cancer. So that's why I was saying it's actually very important to induce these mice early at 6 weeks, because then all of the homozygous mice actually develop cholangio. So obviously we wanted to look at them histologically. It's a really nice model because they actually do go through the stage of Billings towards progression to both intra and extrahepatic cholangio. And we stained for some of the markers that we're supposed to according to the consensus paper. CK19 is high, CK7 is high. We've got stromal deposition illustrated by the picrocereus red for collagen, and also PAS staining. And I should say a big shout out to Guido Carpino here, because he's helped us and looked at all of these tumours in depth. And they recapitulate many of the features of human. So this is the perihiller cholangios, and they recapitulate those features really nicely, both in terms of just H&E, CK19 and PAS staining. The intrahepatic cholangios in the mice actually recapitulate the features of human large duct ICCA. Again, thanks to Guido for comparing these to his human samples. So I'm reliably informed these look a lot like human large duct ICCA. And if we look more closely, so they're CK19 positive, they're CK7 positive, they have high picrocereus red, and they're PAS staining positive. And the tumours appear to be within the larger bile ducts and segmental ducts rather than the small tubules and interlobular ducts. So that was very nice. When we look, obviously these are driven by KRAS, oncogenic KRAS, and P10 loss. So we looked at some of the downstream pathways, and also obviously in humans, these are also prognostic of poor prognosis. And you can see really nicely that we've got high expression of phospho-ERK, downstream of MEK, high expression of phospho-mTOR and of phospho-AKT, which are actually downstream of both P10 loss and KRAS activation. We've done RNA-seq on these tumours and compared to human cholangiocarcinoma, and at a transcriptional level, there really aren't huge differences if you just do a unsupervised clustering analysis. We can look at that more closely. So this is just a UMAP of human and mouse samples from a variety of different tumours. So the black dots here are cholangiocarcinomas. The circles are human, the triangles are mice. They're actually sitting in between HCC and pancreas, which in my head makes sense, just from a geographical point of view. If I zoom in on this, you can see that the mouse and the human are actually clustered pretty nicely together. So what about our other ductal models? I said that we'd done SOX9 Cree-ER mice as well, with the same genetics. So again, deletion of 1 or 2 copies of P10 significantly reduced survival or accelerated cancer development in these mice. Again, they develop both intra and extrahepatic cholangiocarcinoma. The latency is slightly longer, but still reasonable to work with. When you just have P10 wild type, we don't get any tumours in these mice. So again, we looked in the bloods of the mice to see if they resemble the patients. And again, you can see bilirubin, ALK, phos, AST, ALT, bile acids are all up. Albumin is down in the mice, similar to what is seen in the patients, or at least so I'm told. And again, when we look at the histology, we can see that they share histological features with human CCA. We've got both intrahepatic and extrahepatic. Again, we progress through Billings as well. But you can see that they're CK19 positive, CK7, we've got Picrocerius red again. But actually, when we look at the intrahepatics, now in contrast to the CK19 3ER model, which had large duct type tumours, the intrahepatics in this model are small duct type tumours. So they're PAS negative instead of PAS positive. And histologically, you can see that they look much closer to human small duct type. So can we use them for preclinical testing? I mean, that's essentially what we do in my lab. So, and we have a favourite combo in pancreatic cancer. So we thought we would test for that. So what we do, we take tumour-bearing mice from 16 days post-induction, so that's a week before their median survival. So relatively late stage, at least for a mouse. We've done a quick experiment with cisplatin. Bear in mind that I'm not funded to work in cholangiocarcinoma, so we will do GemCis as well. But these are preliminary data, at least for chemo. We got negligible benefit with cisplatin, which is probably not surprising. But when we actually use targeted therapy in these animals, so where we inhibit mTORC1 and 2 and MEK, which are the main pathways activated by these genetic mutations, you can see that we can really nicely extend median survival in the tumour-bearing mice. We got one cured animal here who really should be censored, but died essentially of probably drug toxicity after being on treatment for nearly a year. We've also just started treating the SOX9 model, and you can see again numbers are low, but we're getting nice extensions in survival in that model too. So the target inhibition when we do the treatment experiments, that's maintained. So these are tumours at end stage, and you can see that phospho-ERK remains down, phospho-4-EBP remains down, that's downstream of mTORC1. Phospho-NDRG1, which is downstream of mTORC2, remains down, and that will become significant in a minute. AKT, interestingly, is reactivated, but only in the extrahepatic tumours and not in the intrahepatics. So there's some resistance mechanism there, but it's not the same in intra and extrahepatic disease. What's interesting is that when we use this combo in pancreatic cancer, phospho-NDRG1 stays down in the tumour cells but comes up in the fibroblasts. So it's reactivated in the stroma. And it's possible that in our intrahepatic cholangio we might have a stromal resistance mechanism as well. We just haven't figured it out yet, but I think it is important to pay attention to the stroma as well. So just in summary, I've showed you, hopefully, that CK19 specific KRAS activation and P10 deletion drives cholangiocarcinoma in the mice and recapitulates both extrahepatic and large duct type intrahepatic disease. SOX9 specific activation of KRAS and deletion of P10 also drives cholangio, but that recapitulates small duct type, ICCA, as well as extrahepatic. The CK tumours, we haven't done the SOX9 yet, but the CK model, the tumours are transcriptomically similar to human CCA. Cisplatin has limited efficacy in this setting, but inhibition of both MEK and mTORC1/2 significantly prolongs survival in the tumour-bearing mice. However, resistance develops through alternative mechanisms of AKE activation. That's opposite to what we see in PDAC, and it's intrahepatic, extrahepatic specific. And what we think going forward is that understanding these resistance mechanisms might also be important in the setting of the novel mutant KRAS inhibitors. So that's our future plan, to try and work some of that out. So with that, I would just like to thank Team Pancreas and Curtis, who is Team Cholangio. It's his last day today, and he's having drinks at 7 o'clock, so I'm on the 6 o'clock flight. So if anybody has any burning questions after 5 o'clock, email me. I also want to thank the funders and the Beatson services who make it possible to go rogue and follow up some of these things. And I want to thank Chiara and Guido and Simon at AstraZeneca. And I have to, obliged to point out the excellent Beatson International Cancer Conference in July. And with that, thank you for your attention.